Okay, this time we'll talk about neurotransmitters. What are these substances, and how do they, work at a synapse? Neurotransmitters, first of all, are chemicals made by the neurons. A specific type of neuron produces a specific type of neurotransmitter. The neurons that produce neurotransmitters could be named after the neurotransmitters they produce. Dopaminergic neuron, serotoninergic neuron. That's how you typically name the neurons, adrenergic neuron, etc. So these are small molecules, and typically these are rapidly acting ones. Compared to chemicals like hormones, these are much faster acting. Okay, examples. A very good example is acetylcholine. This is the one that has been studied extensively, the very first neurotransmitter to be discovered. Norepinephrine, dopamine, serotonin, gamma amino, butyric acid, glycine, glutamate, and so on. Neuropeptides are also made by the neurons. Could call them as neuromodulators, more potent than the neurotransmitters. They produce much longer effects, include endorphins, enkephalins. These are opioid peptides, and in other substances, substance P. Diffusible gases are also important as neurotransmitters. Nitric oxide, carbon monoxide are now considered as neurotransmitters. Only neurotransmitters that can also be released by postsynaptic cell to communicate with presynaptic cell. Okay, here are some neurotransmitters that are covered in detail. Acetylcholine has receptors, two types of receptors, nicotinic and muscarinic. We will look at these closely in the autonomic nervous system discussion. They are degraded by an enzyme called acetylcholinesterase. The neurotransmitter that is released at the synapse has to be promptly removed. If it's left behind, it would cause undesired effects at the postsynaptic cell. So it's precisely regulated by the presence of a certain enzyme to degrade the neurotransmitter, or by the uptake of neurotransmitter soon after it's released. And in this case, for acetylcholine, it's the enzymatic action, acetylcholinesterase, is the one that breaks down acetylcholine. Glutamate, glycine, GABA is gamma amino butyric acid. They have different types of receptors, GLU receptors, glycine receptors, GABA receptors. And here it's the reuptake by neurons or glial cells. The serotonin, dopamine, norepinephrine, and epinephrine, all these neurotransmitters, they have a variety of neurotransmitters, 5-HT, D1, D2, alpha-adrenergic, beta-adrenergic receptors, and this is again reuptake is how you degrade them. These are neuropeptides and gaseous type of neurotransmitters. Let's take a look at how these are handled. Receptors are too numerous to list, but are specific to the peptides. Degradation by peptidases, because these are peptides. Peptidase would be the one that breaks it down. Okay, so this is a typical summary of how neurotransmitters are handled, synthesized by the precursors. It could be made in the cell body and stored in the axon terminal, or by the reuptake of the neurotransmitters into the synaptic knob, where the construction can happen locally. Stored in vesicles, an action potential triggers the release of calcium calcium, which then allows the fusion of vesicles with the presynaptic membrane and release neurotransmitters, bind to receptors in the postsynaptic membrane, are deactivated through reuptake of enzymatic degradation. So here is a little bit of detail about glutamate. Glutamate is an excitatory neurotransmitter. It's an amino acid neurotransmitter made of glutamic acid. Okay, it could use either ionotropic or metabotropic type of receptor. Okay, degradation is by the reuptake and repackaging within the presynaptic cell. Reuptake and conversion of glutamine in glial cells is also there, and the glutamine can be converted to glutamate in presynaptic axon terminal. Facilitation. What is this? Typically, an action potential triggers release of same number of synaptic vesicles each time. So anytime there is an action potential, so many vesicles are triggered to release their neurotransmitters. However, a neuromodulator, a diffusible gas, okay, that is released by another cell near the presynaptic cell, okay, could manipulate the activity of the calcium channel, okay, and increase the amount of calcium being released, and therefore more neurotransmitter released. So the effect is bigger than normal. So facilitation is where you're stimulating the presynaptic neuron to bring about a better effect than otherwise. So now let's talk about serotonin 5-HT. This typically uses metabotropic receptor, and so it sensitizes some calcium channels. What that means is if you do this over a long period of time, release of serotonin over a long period of time, this would become a regular situation. That is, calcium channels would open with the lower threshold. So basically, it's very easy to open calcium channels, and they move a lot of calcium into the cell, and therefore there's increased release of the neurotransmitters. Arriving APs thus open more calcium channels. More calcium means more vesicles unbound from cytoskeleton and more neurotransmitters released. We already know about the recycling of neurotransmitters, either enzymatic breakdown, reuptake, or diffuse away from the synaptic cleft.